CLL is a rather special malignancy. Lymphomas in general are special because they originate from mature B cells and from mature cells, mature lymphocytes. Lymphocytes are engaging in communication with their environment, and so it was not paradoxical, rather the contrary, in fact, that mature lymphomas of mature cells, and today we are talking about a lymphoma of a mature B cell, because chronic lymphocytic leukemia is exactly that. They are also engaging in interactions with the microenvironment. They are receiving signals of various kinds. And they do have one receptor in particular that is implicated in recognition of specific signals. They are called antigens. This is the B cell receptor. But there is also a plethora of other receptors that are triggered by external stimuli. Now, this external stimulation appears to be highly important for both the proliferation of the malignant cells, but also their resistance to death. So they have a prolonged lifespan, but they proliferate more when they receive the signals. And this is extremely important because we have found ways of interfering with the process of activation. So we are blocking the signaling, the pathways that we mentioned, the signaling pathways, and this has translated into very, very remarkable responses, even in patients refractory to many other forms of treatment. So this is a new concept in the management of patients with CLL and other lymphomas, I should say. So this is a major, a hot topic these days. But what we also learned today is that this specific receptor, the B cell receptor, is not alone. And many speakers, some of the speakers in that session, highlighted the importance of other pathways that seem to be, of course, complementing, or I should say interacting also with the B cell receptor. So it's not a simple story. It will never be that there is a single effect or a single pathway that is disturbed and you would be doing this or that and cure the disease. It will be far more difficult than that. So this was one part of today's session. The other was that we understood, we have new information about how this communication with the external environment is taking place, because the process has been now dissected in great detail at the molecular level, and we are hopeful that all this new knowledge will assist in the design of better therapies.